Now we will start to look at muscle tissue and how muscle cells work at the cellular level. This portion of the muscular system focuses largely on the physiology of muscle contraction, which is often a place where students struggle. If you're having trouble, be sure to ask for help right away. Let's start by looking at how our skeletal muscle tissue is organized. If we take a piece of our skeletal muscle and look at it on cross-section, there are 3 layers of connective tissue that surround the muscle. The outermost layer is the epimysium. This is a dense layer of collagen fiber that covers the entire muscle. Between the bundles of muscle fibers or fascicles is the perimysium. The perimysium consists of collagen fibers, blood vessels, and nerves. Lastly, the surrounding individual muscle fibers is the endomysium. This consists of elastic fibers, capillaries, and nerves. Let's take this muscle and look at it a little more closely in cross-section. I created this image using AI to better demonstrate muscle structure. If we take the muscle in cross-section and telescope it out a little bit, we can take a better look at these connective tissues. Again, the epimysium covers the entire muscle. The perimysium covers groups of muscle cells called fascicles. Endomysium covers individual muscle cells. The tendon that holds the muscle to the bone is where these 3 connective tissue layers come together. This tendon is a rope-like tendon. However, tendons can also be flat. A flat tendon is referred to as an aponeurosis, like the gala aponeurotica that connects the frontalis muscle to the skull. Now we move on to look at the structure of individual muscle cells. This is where the magic of muscle contraction actually takes place. As we go through these structures, keep in mind the word root myo means muscle and sarco means muscle. A single muscle cell can also be referred to as a myofiber or muscle fiber. The plasma membrane that surrounds each muscle cell is the sarcolemma. Inside each muscle fiber are many organelles called myofibrils. Inside the myofibril are the myofilaments. These filaments are made of actin and myosin proteins, and it is here that muscle contraction occurs. We will talk about the process of muscle contraction a little bit later. Let's look at additional structures within the muscle cells that contribute to muscle contraction. Each myofibril is wrapped in the sarcoplasmic reticulum, or SR. This structure is like a smooth endoplasmic reticulum. The sarcoplasmic reticulum stores calcium ions that are needed for muscle contraction. Traveling through muscle cells are these structures called T-tubules or transverse tubules. They are connected to the sarcolemma and are important for carrying an electrical signal from a nerve cell to the sarcoplasmic reticulum to release calcium needed for muscle contraction. At the ends of the sarcoplasmic reticulum are the terminal cisternae. These are fattened areas of the SR that contain a large amount of calcium ions. 1 T-tubule and 2 terminal cisternae are referred to as a triad. It is here that the electrical signal in the T-tubule stimulates the release of calcium from the SR. Now let's look more closely at the myofilaments and how they function. The myofilaments are part of the myofibrils. Myofilaments are bundles of thick and thin filaments. These filaments are organized into a sarcomere. The sarcomere is the functional unit of the muscle cell. The functional unit is where muscle contraction takes place, and it happens due to the interaction of the myosin and actin filaments. The sarcomere is organized into region. This region here is called the I band because it's light in color when viewed under a microscope. This band here is the A band because it is dark in color when viewed under the microscope. The alternating I and A bands is what gives skeletal muscle the striated appearance when viewed under the microscope. The thick filaments are made of myosin proteins, and the thin filaments are made of actin proteins. The Z line is where the actin filaments are anchored, and the myosin proteins are also anchored to the Z line with stabilizing proteins called titin. The H band, or H zone, is this region that contains thick filaments but no thin filaments. The zone of overlap is where the actin and myosin filaments overlap. Let's look more closely at the structure of thin filaments. While it is largely made of actin proteins, there are other important proteins that contribute to making this filament. These are troponin and tropomyosin. Tropomyosin is a filamentous protein that covers active sites on actin. The active site is where the myosin protein interacts with actin during muscle contraction. When the muscle is not contracting, tropomyosin covers the active site and is held in place with troponin. When we discuss muscle contraction in a little bit, you will begin to understand how these proteins are related to that process. The thick filaments are made of myosin proteins. Myosin proteins have a head region and a tail region. The head region is what interacts with the actin filaments during muscle contraction. The theory of muscle contraction is known as the sliding filament theory. In this theory, the thin filaments will slide toward the center of each sarcomere alongside each thick filament. This will cause the sarcomere to shorten, which leads to muscle contraction. Here is a sarcomere and a relaxed muscle. Note the size of the A band, I band, H zone, and zone of overlap. In contrast, here is a sarcomere and a contracted muscle. What has happened to the size of the I band, A band, H zone, and zone of overlap? Notice that as the sarcomere shortens during contraction, the A band remains constant, the I band gets smaller, the H zone gets smaller, and the zone of overlap gets larger. These changes support the idea that shortening of the sarcomere occurs as actin filaments slide past the thick filaments toward the center of the sarcomere. Shortening of the sarcomeres that make up the myofilaments within the myofibrils causes shortening of the muscle cell and in turn contraction of the muscle. Before we move into the details of muscle contraction, it's important to understand the key principles governing muscle contraction of the skeletal muscle cells. The first thing you absolutely need to remember is that when muscles contract, they pull on a tendon. Muscles never push on anything. They always pull on the attached tendon and as a result moves the bone or tissue it is connected to. Secondly, contraction only occurs when muscle fibers are stimulated to contract at the neuromuscular junction. The neuromuscular junction is where the nervous system, neuro for nerve, meets the muscle. The third thing is that the tension produced is dependent on the number of motor units stimulated. A motor unit is the neuron and the muscle fibers it stimulates. This means that a single neuron can stimulate many muscle cells. Motor units can vary in size from small to large. The size of the motor unit relates to how well that movement is controlled. Small motor units are made of one neuron controlling a few muscle fibers and allow for fine motor control. Fine motor control is important for precise movements like those that occur when moving the eyes. Large motor units consist of one neuron controlling 1,000 or more muscle fibers. This type of motor unit is more common in areas where fine motor control is not as important, such as when contracting muscles in the leg. A good analogy to help you understand the difference between these motor units is the management of workers. If you are a manager of 3 or 4 employees, it's easy to make sure these individuals are completing the job as required. However, if you are a manager of several thousand people, it becomes a lot harder to make sure these individuals are completing the job as required. There is less control with a large motor unit compared to a small motor unit. Now we are moving on to start discussing the physiology of muscle contraction and explore what happens when a neuron stimulates a muscle cell to contract. We start with the events that take place at the neuromuscular junction, where the motor neuron stimulates the skeletal muscle cell to contract. This neuron is called a motor neuron because it targets muscle cells specifically. There is a lot that takes place in this very small space. It starts with an electrical signal that travels down to the end of the motor neuron. This electrical signal is also known as an action potential. When the action potential arrives at the end of the motor neuron or axon terminal, it stimulates the opening of calcium channels. Remember channel-mediated transport discussed during the cellular level of organization? This is an example of where that type of transport is important for cellular function. As the calcium ion channels open, calcium ions will move into the axon terminal from an area of high concentration to low concentration. The entry of calcium ions stimulate the movement of vesicles containing the neurotransmitter acetylcholine to move toward the membrane, fuse, and discharge their contents into the small space between the axon terminal and the muscle cell called the synapse. What kind of cellular transport does this represent? If you said exocytosis, you're correct. If you could not remember, you should review the cellular transport mechanisms discussed earlier in this course. Once acetylcholine is released, it travels across the synaptic cleft to receptors on the surface of the muscle cell membrane. The receptor is another channel protein, which then opens and allows sodium ions to enter the muscle cell. They are also moving from an area of high concentration to low concentration. This movement of sodium into the muscle cell triggers an electrical signal that then travels across the sarcolemma and enters the T-tubules. Use this slide to review the steps just covered to label the diagram. Try to complete this without the recording for help. Once you are finished, rewatch the recording to check your work before moving on to part 2.